Okay. Here we go. And so let's begin. Hello. It's me again, Dmitry. I haven't done one of these recently. There's been a lot of other things going on with my academic work, so I haven't done one of these recently. But I am back to reading the physiology textbook today, and we'll be going over chapter 33 this time. I'm sure you've all missed me and missed these physiology textbook chapters, so we'll get right into it. Chapter 33, Red Blood Cells, Anemia, and Polycythemia. A major function of red blood cells, RBCs, is to transport hemoglobin, which in turn carries oxygen from the lungs to the tissues. Normal RBCs are biconcave discs, though the shapes can change markedly as the cells pass through the capillaries. A normal RBC has a great excess of cell membrane relative to the quantity of material it contains. Deformation of the cell does not stretch the membrane and consequently does not rupture the cell. The average number of RBCs per cubic millimeter is 5.2 million plus or minus 300,000 in men, and 4.7 million plus or minus 300,000 in women. Red blood cells have the ability to concentrate hemoglobin. In normal individuals, the percentage of hemoglobin is almost always near the maximum level in each cell, about 34 grams per deciliter. The blood contains an average of 15 grams of hemoglobin per 100 milliliters, 16 grams in men and 14 grams in women. Each gram of pure hemoglobin is capable of combining with approximately 1.34 milliliters of oxygen. In a healthy person, more than 20 milliliters of oxygen can be carried in combination with the hemoglobin in each 100 milliliters of blood. Genesis of blood cells. All circulating blood cells are derived from pluripotential hematopoietic stem cells. The pluripotential cells differentiate to form the peripheral blood cells. As these cells reproduce, a portion is exactly like the original pluripotential cells. These cells are retained in the bone marrow to maintain a constant supply. The early offspring of the stem cells cannot be recognized as different types of blood cells, even though they already have been committed to a particular cell line. These cells are called committed stem cells. Different committed stem cells produce different colonies of specific types of blood cells. The growth and reproduction of the various stem cells are controlled by multiple proteins called growth inducers, which promote growth but not differentiation of the cells. Differentiation is the function of another set of proteins called differentiation inducers. Each of these inducers causes one type of stem cell to differentiate one or more steps toward the final type of adult blood cell. The formation of growth inducers and differentiation inducers is controlled by factors outside the bone marrow. In the case of RBCs, exposure of the body to a low level of oxygen for a long period induces growth, differentiation, and production of greatly increased numbers of erythrocytes. Erythropoietin regulates red blood cell production. The total mass of RBCs in the circulatory system is regulated within narrow limits. Any condition that causes the quantity of oxygen that is transported in the tissues to decrease ordinarily increases the rate of RBC production. The principal factor that stimulates RBC production is the circulating hormone erythropoietin. In a normal person, about 90% of erythropoietin is formed in the kidneys, with the remainder formed mainly in the liver. The structure in the kidney in which the erythropoietin is formed is not known. Some studies suggest that erythropoietin is secreted by fibroblasts-like interstitial cells surrounding the tubules in the cortex and outer medulla, where much of the kidney's oxygen consumption occurs. Other cells, including the renal epithelial cells, also create erythropoietin in response to hypoxia. When both kidneys are surgically removed or destroyed by renal disease, the person invariably becomes extremely anemic because the amount of erythropoietin formed in non-renal tissues is sufficient. is not, it's not, let's see, is sufficient to cause only one, that's an informed, sorry. Let me read this. That's a very funny cutoff point. Okay. Let me read that again. When both kidneys are surgically removed or destroyed by renal disease, the person invariably becomes extremely anemic because the amount of erythropoietin formed in the non-renal tissues is sufficient to cause only 1/3 to 1/2 as many RBCs to be formed as are needed by the body. That's a big difference. I.e., they're not sufficient. They're not sufficient to actually supply the body in a normal sense. I.e., they're going to be anemic. And so here's this chart right here. You can see hematopoietic stem cells, which are going to be under the influence of erythropoietin to become pro-erythroblasts, and then they're going to become RBCs, and they're going to promote tissue oxygenation, which is going to be a negative feedback loop on the production, or I should say the release of erythropoietin from the kidneys. Likewise, factors that decrease oxygenation, so that's like low blood volume, anemia, low hemoglobin, poor blood flow, pulmonary disease, etc. These are going to obviously decrease tissue oxygenation and therefore are going to increase erythropoietin and therefore obviously produce more RBCs. So that's kind of how that works. Very simple. So let's proceed. Final maturation of RBCs requires vitamin B12 and folic acid. Both vitamin B12 and folic acid are essential to the synthesis of DNA. The lack of either of these vitamins results in a diminished quantity of DNA and consequently failure of the nuclear maturation and division. In addition to failure to proliferate, the RBCs become larger than normal, developing into megaloblasts. These cells have irregular shapes and flimsy cell membranes. They are capable of carrying oxygen normally, but their fragility causes them to have a short lifespan, 1/2 to 1/3 that of normal. Vitamin B12 or folic acid deficiency therefore causes maturation failure during the process of erythropoiesis. A common cause of maturation failure is an inability to absorb vitamin B12 from the GI tract. This inability often occurs in persons with pernicious anemia, a disease in which the basic abnormality is atrophic gastric mucosa. The parietal cells of the gastric gland secrete a glycoprotein called intrinsic factor, which combines with the vitamin B12 to make it available for absorption by the gut. The intrinsic factor binds tightly with vitamin B12 and protects the vitamin from indigestion by the GI enzymes. The intrinsic factor vitamin B12 complex binds to a specific receptor sites on the brush border membranes of mucosal cells of the ileum, so that'd be in the small intestine. Vitamin B12 is then transported into the blood via the process of pinocytosis. A lack of intrinsic factor causes a loss of much of the vitamin resulting from enzyme action in the gut and failure of absorption. Very important to note. And then it's interesting, goes back into a general concept about how, because of the interconnectedness of the body, how something as simple as how the parietal cell is not functioning correctly and they can't produce intrinsic factor in the stomach can lead to something like your blood cells being dysfunctional because you no longer have B12 absorption. So, hemoglobin formation. Synthesis of hemoglobin begins when the RBC is in the pro-erythroblast stage and continues into the reticulocyte stage, at which point the cell leaves the bone marrow and passes into the bloodstream. During the formation of hemoglobin, the heme molecule combines with a long polypeptide chain called a globin to form a subunit of hemoglobin called hemoglobin chain. 4 hemoglobin chains bind together loosely to form the entire hemoglobin molecule. The most important feature of the hemoglobin molecule is its ability to bind loosely and reversibly with oxygen. The oxygen atom binds loosely with one of the so-called coordination bonds of the iron atom in hemoglobin. When bound to the iron heme, oxygen is carried as a molecular oxygen composed of 2 oxygen atoms. So that's O2, right? Oxygen is released into the tissue fluids in the form of dissolved molecular oxygen rather than as ionic oxygen. So true. Iron metabolism. Iron is important for the formation of hemoglobin, myoglobin, and other substances such as cytochromes, cytochrome oxidase, peroxidase, and catalase. The total average quantity of iron in the body is about 4 to 5 grams. About 65% of this amount is in the form of hemoglobin. About 4% is in the form of myoglobin. Let me read that again, actually. Waiting for something to load correctly. As I was saying, about 4% is in the form of myoglobin. 1% is in the form of the various heme compounds that promote intracellular oxidation. 0.1% is combined with the protection transferrin in the blood plasma, and about 15 to 30% is stored mainly in the reticuloendothelial system and liver parenchymal cells, principally in the form of ferritin. And that's basically the storage form. Transferrin is like a transport form, and then ferritin is going to be like more of a long-term storage form comparatively. Iron is transported and stored. When iron is absorbed from the small intestine, it almost immediately combines with a beta globulin called apotransferrin to form transferrin, which is transported in the plasma. This iron is loosely bound. Excess iron in the blood is deposited in liver hepatocytes and in reticuloendothelial cells of the bone marrow. Once inside the cell's cytoplasm, iron combines with the protein apoferritin to form ferritin. Varying quantities of iron can combine in clusters of iron radicals in the ferritin. So just be aware, basically, the complex between the apoferritin and the iron is called ferritin, and the complex between the iron and the apotransferrin is just called transferrin. So the apo form means you're lacking the iron. But when you add the iron to that apo molecule, either it be apotransferrin or apoferritin, you drop the apo part and just called transferrin or just called ferritin. FYI, if you see it on the exam or something like that, just be aware of the difference. When the quantity of iron in the plasma decreases to less than normal, iron is removed from the ferritin quite easily and transported by transferrin in the plasma to the portions of the body where it is needed. A unique characteristic of the transferrin molecule is its ability to bind strongly with receptors in the cell membranes of the erythroblast and bone marrow. Transferrin is ingested by endocytosis into the erythroblasts along with the iron that is bound. Transferrin delivers the iron directly to the mitochondria, where the heme is synthesized. When RBCs have reached the end of their lifespan and are destroyed, the hemoglobin released is ingested by cells of the monocyte macrophage system. The free iron that is liberated can be stored in the ferritin pool or reused for formation of hemoglobin. Anemias. Anemia means a deficiency of RBCs and can be caused by a rapid loss of RBCs or slow production of RBCs. Blood loss anemia occurs after significant hemorrhage. The body is able to replace the plasma within 1 to 3 days. However, the concentration of RBCs remains low. After significant hemorrhage, a period of 3 to 4 weeks is required for the number of RBCs to return to normal levels. Aplastic anemia is the result of non-functioning bone marrow, which may be due to exposure to gamma radiation for cancer treatment or toxic chemicals such as insecticides or benzene and gasoline. Autoimmune disorders such as lupus erythematosus result in an immune system attack on the healthy cells of the bone marrow, which destroys stem cells and may lead to aplastic anemia. Individuals with severe aplastic anemia usually die unless they are treated with blood transfusions or bone marrow transplants. Megaloblastic anemia is the result of a lack of vitamin B12, folic acid, or intrinsic factor, which obviously if you're lacking intrinsic factor, it also means you're going to lose B12. So it's kind of a downstream effect of that. Lack of these substances leads to the slow reproduction of the erythrocytes in the bone marrow. As a result, these erythrocytes grow into large, odd-shaped cells called megaloblasts. Hemolytic anemia is the result of fragile RBCs that rupture as they pass through the capillaries. With hemolytic anemia, the number of RBCs that form is normal or in excess of normal. However, because these cells are extremely fragile, their lifespan is very short. Sickle cell anemia is a type of hemolytic anemia caused by abnormal composition of the globin chains of the hemoglobin. When this abnormal hemoglobin is exposed to low concentrations of oxygen, it precipitates into long crystals inside the RBC, which cause the cell to have an abnormal sickle shape and to be extremely fragile. And you might ask some questions like, well, what situation would you have a low oxygen environment? Well, there'll be two situations. One, the person themselves with sickle cell disease is in fact in a low oxygen environment. Let's say, for example, they are hiking on a mountain. Well, that can trigger it. That's one thing. The other thing is that simply in the body itself, your blood cells are going to be exposed to low oxygen environments sometimes because they're traveling from the heart through the arteries to the capillaries where they're losing all this oxygen, they're giving away this oxygen, and then they're coming back through the veins. So both in the context of simply how the body circulates blood, it can lose oxygen contextually, depending on where it is. If it's already given up its oxygen, it could potentially sickle in that situation. But also, if you're in a low oxygen environment externally because you're hiking on a mountain or something like that, that can also trigger it. Polycythemia. Polycythemia is a condition in which the number of RBCs in the circulation increases hypoxia or genetic aberration. Let me read that again. Polycythemia is a condition in which the number of RBCs in the circulation increases, hypoxia or genetic aberration. I think what they mean is that polycythemia is we have an elevated number of RBCs, and that it could be caused by hypoxia or genetic aberration. I think that's what they mean by that. Individuals who live at high altitudes have physiologic polycythemia as a result of the thin atmosphere. Yeah, basically what they're saying is, again, polycythemia means you have elevated RBCs, elevated red blood cells. And the reason is because there's something like, for example, physiologic polycythemia because you have high altitudes. So that's where you're going to have a hypoxic situation where you want to, sort of compensate for that by increasing your RBCs. Or it could be because of a genetic aberration. Polycythemia can occur as well in individuals with cardiac failure because of decreased delivery of oxygen to the tissue. So basically the body's trying to compensate again for low oxygen delivery, or is trying to at least. Polycythemia vera is a genetic aberration in the hemocytoblastic cell line. The blast cells continue to produce RBCs even though too many blood cells are present in the circulation. The hematocrit can rise to 60 to 70%. Because polycythemia greatly increases the viscosity of the blood flow through the vessels is often sluggish. Very true. And this kind of shows that even in a situation where you would think, how could high RBCs be a problem? Well, in this case, if you really have this elevated level, it could cause the blood to actually become sort of sluggish because it's so viscous from the RBC. So there's really there's a reason why there's always a balance to things, Anyway, that concludes Chapter 33 of Guyton Hall's Pocket Physiology textbook. You can follow along in the Pocket Physiology textbook, or if you really want to, you can read it in the normal Guyton Hall textbook. But that would be the end of our chapter. Thank you for watching.